Okay, fine. We are continuing with the topic which we had discussed in our last session, that is solute transport and cell organelles and protein targeting. Okay. Solute transport, I won't be taking much questions because majority of them I have taken it last time. So mainly my focus will be on cell organelles and protein targeting. But firstly, few questions from solute transport. So here is the first question to you. Cystic fibrosis transmembrane conductance regulator. That means we are having CFTR channel. It is known to be a CAMP dependent chloride channel. CF patients, that is cystic fibrosis patients, show reduced chloride permeability and as a result exhibit elevated chlorine levels in sweat. Now, to prove this, CFTR proteins, both wild type and mutant, are inserted in a model membrane, that is liposome. I hope so you all are aware of liposomes. And chloride transport is followed with radioactive chloride ions. Now, it is known that topology of CFTR in membrane is very important for its function. So, despite not undergoing proteolytic degradation or denaturation, wild-type CFTR fail to transport chloride in the liposome. Now, you have to identify the correct explanation for this. The reason is CFTR gets mutated during insertion in liposomes or loses its affinity with chloride ions, gets wrongly inserted within the liposomes, or loses the channel forming property in the liposomes. Find out the correct answer over here. Very good evening, Sunshine, Rahul, Anusha, Kumar, and Kirtana. So you have to have an idea about CFTR channel. Along with that, you should have an idea about liposomes. So combining the two concepts together, you have to get the answer. What is the reason, even though CFTR proteins are not undergoing denaturation or degradation, wild-type CFTR failed to transport chlorides in the liposomes? What should be the answer over here? Anyone? You understood the question, right? They are just asking what is the reason that CFTR is not able to transport chloride ions when we are taking a liposome. Liposomes means what? These are artificial lipid vesicles. Commonly used for what? Commonly used for transportation of drugs, many other components like biomolecules, etc. What should be the answer? Good evening, Neha. I can see some miscellaneous answers are coming. Saujanya, Rahul, Sunshine, and Neha. Anyone else would like to give the answer? Just understand the connecting link between CFTR protein to that of a liposome. If you are able to correlate that, then you will be able to get the answer. Okay, let me tell you the answer. The correct answer over here is CFTR protein gets wrongly inserted inside the liposomes. What I told you, liposomes means they are responsible for delivery of specific components. And in this case, they are responsible for the transfer of chloride ions. Right. So, if it gets wrongly inserted within the liposome, what happens? Definitely, it is going to affect the protein folding and all. So, if protein is not folded properly, then how the protein will be functioning? See, CFTR, we know that is a protein that functions as a channel in the cell membranes, regulating the movement of ions across the membrane, that we are aware. Then what about liposomes? These are artificial structures made up of lipids that can encapsulate various substances, which can also include proteins. So if the CFTR protein gets wrongly inserted inside the liposomes, it will not function properly. Why? Because liposomes will not be able to provide the necessary environment for the protein to carry out the function. That is first point. Second point is that it will interfere with normal folding and assembly of CFTR. As we all know that folding plays a very important role in protein function, any protein function. So if it interferes with this folding and assembly, what happens? It will definitely affect the functioning of proteins. Okay, so that's why the answer should be 3. I hope so you got the answer. You understood how exactly it works, right? Moving on to the next question. So you can expect experimental questions from Solute Transport. So this is yet another example. A diabetic patient has a high blood glucose level due to reduced entry of glucose into the peripheral tissues in addition to other reasons. But there is no problem of glucose absorption, however, in the small intestine of these patients. So following observations were found. Glucose is transported into the cells of muscles by GLUT transporters, which are influenced by insulin receptor activation. Second, glucose transporters can enter inside the enterocytes, which can be mediated by SGLTs, that is not dependent on insulin. Statement C. Glucose molecules are transported in the small intestine by facilitated diffusion. And lastly, the secondary active transport of glucose occurs in muscles. Now, in this question, what you need to find? You need to find the incorrect combination of statements. So, when you study the question, study it thoroughly, whether they are asking correct combination or they are asking the incorrect combination. So, currently, they are asking incorrect combination of statements. Okay, find out. Here one point need to be considered it. There is no problem of glucose absorption. Glucose absorption is not a problem. But still the person is suffering from high blood glucose level. So among this, which one is the incorrect combination of statements? Just, I think we had discussed GLUT transporters in my last session. Try to correlate, you are able to remember or not. In case, one more tip over here, in case if you are not able to identify, like you are having some confusion, recollect the concept behind correct statements. Okay, so in that way also you can get answer over here. Like there will be certain statements which you will be confident, no, this is the correct answer or this is the correct statement. Pick up that and check out the options and see whether it is possible or not. Yes, Rahul has given the answer. What about others? Others. Okay, Surinder Rahul has given the answer. Try out everyone. Very good question. And these kind of disease related or experimental questions you can often expect in CSIR exam. Okay, you are having confusion between 2 or 1. Okay, fine. Ashu, Saujanya, Deepshika. Okay, I can see majority of you are saying 4. Again, some combination of 3 and 2 is also coming. Let's check it out. What is the correct answer over here? So here what they are asking, incorrect combination of statements over here. So the correct answer is C and D. Now check out A and B. You remember me saying under last class where insulin comes and binds to the receptor, and at that stage, which GLUT transporter is activated in muscles? Insulin when goes and binds to the receptor, at that stage, which glute transporters are activated in muscles? Which glute transporters? You know, we are having different types of glute transporters present in different parts of our body. Glute 1, glute 2, 3, 4, 5, 6. So which one is exclusively working in muscles? Absolutely correct. Glute 4. So this is the correct statement. Glucose is transported into the cells by glute 4 transporters, which are influenced by insulin receptor activation. Now, statement B. Glucose transport into enterocytes can be mediated by SGLTs that are not dependent on insulin. Now, what are the sodium-dependent glucose transporters? What kind of transporters are these SGLTs? So, obviously, if statement A is correct, statement C will be incorrect. They are transmuted in the small intestine by facilitated diffusion. You have to specify whether it is a carrier protein or a transporter. So statement A becomes the correct one. Next. Yes, what kind of co-transport mechanism is that? Is it a symport or an antiport? SGLTs are symport or an antiport? Glute 4 should be the answer. Okay, yeah. Co-transporter is correct. Symport or antiport? Yes. So this becomes a symport mechanism which is not dependent on insulin. So that becomes the correct answer. And secondary active transport does not take place in muscles. That is also the incorrect statement. So if you see over here, we have some examples of symporters like SGLT1. These two exclusively working in small intestine. Sodium iodide symporter in thyroid gland as well as mammary gland. Sodium potassium chloride co-transporter present in loop of Henle. So somewhere these transporters are working where your majority of these transporters or symporters will be working in kidney, or I can say small intestine. Same is the case with sodium chloride co-transporters and potassium chloride transporter. Similarly, we have some examples of antiporters. Sodium calcium exchanger. Where exactly this will be working? Sodium calcium exchanger. Where exactly it will be working? In which tissues or in which organ? Of course, I can say that you might be aware of chloride bicarbonate exchanger, anion exchanger, right? What about this sodium calcium exchanger? It works in, yes, it works in cardiac muscles. Specifically for maintaining cardiac action potential. The way you have neuronal action potential, your cardiac muscles are also having a specific kind of action potential. Then sodium hydrogen exchanger, they are working in kidneys. And any mutation in this exchanger will result in kidney failure. Okay, so these are some examples of symporter and antiporter. Fine. Moving on to the next question. Which of the following describes an amphisome? It is an intermediate organelle produced through the fusion of endosomes with autophagosomes within the cells. A double membrane sequestering vesicle considered to be the hallmark of intracellular catabolic process known as macroautophagy. Compartment formed when autophagosome fuses with the lysosomes, or is a vacuole that arises when membranes of ER sequester parts of the cytoplasm. What should be the answer over here? PCT is also present, Saujanya, proximal convoluted tubule. Where it is present? Again, it is present in kidney only. So that's why I just said as commonly present in kidney. Yeah, regarding this amphisome, again I had discussed in details in my previous session. Okay. Regarding this amphisome, if you are able to recollect, I have given you a diagram and also I have involved the steps which leads to the formation. Does anyone remember what should be the correct answer over here? Yes, very good. Kirtana, Surinder, Sunshine, Ashu, and Anusha. Anyone else would like to give the answer? Anyone else would like to add on? What is an amphisome? Basically, this is an intermediate organelle. Okay? Yes. So, the statement 1 will be the correct answer. It is an hybrid organelle produced through the fusion of endosomes with autophagosomes within the cells. Details I had provided you in the previous session. Okay. Next, autophagy related questions are coming very frequently in the exam. See, another question. Which one of the following statements correctly represents the event shown below? You can see some proteins are present, some images are being displayed over here. So you have to identify which one is the correct answer. The cell is undergoing apoptotic cell death with the help of lysosomes. That means they are saying A is the lysosome. 2. The cell is undergoing autophagy by the formation of autophagolysosomes. They are saying C is autophagolysosome. Cell is undergoing necroptosis. Cell is undergoing autophagy and fusion occurs between lysosome and autophagolysosome. That means A and B are autophagolysosome and lysosome they are saying. So among this, which one should be the correct answer? See, even if you are not aware of these proteins, whatever is mentioned over here, like autophagy 12, bacillin 1, LC32, even if you are not aware, but still you will be able to give the answer. Again, as I told you, like in my previous session, I had mentioned you about the autophagic diagram and all, right? The same thing only being asked over here. I can see miscellaneous answers are coming up. See the image. Remember the sequence of events that occurs during autophagy. Then you will be getting the answer. And read the options properly. See, whenever we are in the exam hall, when we see a question which is quite familiar to us, we become too much of excited. And because of that, we sometimes miss few contents in the question and we may face some negative marking over there. So that's the reason it is mandatory to go through the questions thoroughly. So what should be the answer? Majority of you are saying 4. Anyone else would like to give your inputs? Remember the sequence of events that occurs during the autophagy, you will be able to get the answer. What is this structure? What is this structure known as, the one which I am indicating, this one? What is this structure known as? We have a name for this structure. What is that structure name? Anyone remembers? We had discussed many times in the class and also in the previous session. Okay, if I say this is phagophore, then are you able to identify what does A, B, and C stands for? If I say this structure as a phagophore, are you able to identify what does A, B, and C indicates? You are correct, Surinder. Yes. The correct answer over here is option 2. Why? Because the phagophore will form autophagosome. right? Then lysosome is added to that, right? After that, you are getting a autophagolysosome, this structure, which releases its hydrolytic enzymes for the degradation. Are you getting me? Yes, you are absolutely correct, Surinder. Kirtana, absolutely correct. See, now regarding these proteins, what does they indicate? ATG indicates autophagy. Autophagy 12 is a part of a protein conjugate. We call it as 12 and 5. That means autophagy 12 and 5 forms a conjugate. And what is the function of this conjugate? Helps in the formation of autophagosomes. That is what I said you over here. This structure will be a autophagosome. So this is the phagophore. That means the membrane donated by the ER. Slowly, gradually, it becomes a vesicle, which is known as autophagosome. So your autophagy 12 along with 5 forms or helps in the formation of this structure. Then these are double membrane structures that sequester cellular compartments and deliver them to lysosomes for degradation. Obviously, Beclin 1. Beclin 1 is a protein involved in the initiation of autophagy. In fact, they are responsible for the recruitment of other proteins involved in autophagy. That means you are having different types of autophagy proteins, 12, 5, 4, like that. So those recruitment is carried out by Beclin 1. And the last protein is LC32. What is this? LC32 is a protein that is involved in the formation of, again, autophagosome. So remember, this protein and this protein contributes to the structure of autophagosomes. Beclin is responsible for recruitment of other autophagic proteins required to carry out autophagy. Okay, I hope so. This is clear to everyone. No confusion over here. No confusion over here, right? The question and the image and the answer, you are able to correlate, everyone? Yes, we can proceed with other question now. Next question. Clear? Okay, very good. Next. To delineate the steps in endoplasmic reticulum transport, a PhD student homogenized pancreatic acinar cells to isolate microsomes, which retain most of the biochemical properties of endoplasmic reticulum. For this experiment, the student has planned a number of controls as mentioned below. Treat one set of microsomes, first with detergent, then with proteases, or treat the set of microsomes with protease only, or treat it with micrococcal nucleases, or treat it with detergent only. Now you have to identify the correct combination of control which that student has taken. So here are the options for you: A, B and D, B, C and D, A, C and BD. Read the statements properly and identify which can be used as a control. Identify the statements and find out. So what is a microsome? Microsome is, are you aware of this term microsomes here? What it is? Or from where we get this microsome? Something related to endoplasmic reticulum? Yes? Is it tough to answer? Yes. Derived from endoplasmic reticulum in the form of a vesicle. Okay. Now you are able to identify which one can be used as a control by this PhD student. in order to understand the steps in ER transport? A, B, and D mentioned by Rahul. Okay. What about others? Try on. Definitely you will get. Just read the question properly. Understand the function of enzymes. You will definitely able to get the answer. Even you don't require the combination as well. You can say directly. Protease is there. Okay, fine, Kirtana. Okay, Sunshine. Others, I can see very less response for this question. Is it a tough question? Even though it looks tough, but when you know the answer, you will see that it is an easy question. And this is a 4 marks question, came previous to previous year in the CSIR. Okay, let me give you the answer. What does it says? The correct answer over here, as mentioned by Rahul, A, B, and D. Now, how A, B, and D is there? C. Simply by looking into this option, you can eliminate statement C. Why you can eliminate statement C? Because nuclease is an enzyme responsible for the degradation of nucleic acids. And here we are discussing about microsomes, which is having no connecting link with nucleic acid. So that's why you can easily eliminate these options, this statement. So the microsomes are heterogeneous set of vesicles formed from ER when our ER cells are disrupted. And these vesicles can be obtained by differential centrifugation and comprises of 3 features: rough vesicles, smooth vesicles, and separately the ribosomes. In fact, microsomes are the most widely used preparation of ER membrane, commonly used for co-translational processing of membrane proteins. You know, we are having 2 types of transportation, co-translational and post-translational. So this is used for co-translational processing. Now, detergents are used to lyse the cells, releasing soluble proteins. So membrane proteins and lipids can be removed. Proteases, as you said, yes, it will be acting upon the peptide bonds of protein. So in control experiment, all the 3 can be used. That is detergents, proteases, and what was the other one? The combination as well. Individually as well as in combination. But nucleases, specifically, you are using it for nucleosome positioning and chromatin structure. That means something that is related to our nucleic acid. So we can easily avoid that nucleus, switch on to other 3 components. Okay, so this is how you have to interpret the answer. I hope so this is also clear to all of you. No confusion over here. If you come across these kind of questions, try to eliminate the option. In case if you are not able to get the answer, what you can do, find out the enzyme's function and eliminate it. Once you eliminate, you'll be able to get the answer. Because this is a kind of generalized question where you don't have to remember the concept and all. The ER transport and all. Those are not required. Just a, what you can say, basic idea of enzymes. Okay. Okay. So next question. Okay, before moving to the next question, just wanted to remind you about the Watson Impact Scholarship, which is exclusively for biotech graduates, postgraduates, and professionals. Through this, one can avail different kinds of scholarship. That means you can start from 25% to 50%, even one can go for 100% scholarship as well. And what all things you can get it under scholarship? You can go for attending workshops, webinars, mentorship programs. Even you are having some smart tools that will help you in revising the concept quickly. So these are something that comes under the Y scholarship. Now, If you want to know further details, just dial to these numbers. You will get more information regarding the same. And remember, the deadline is 31st July. So very few days to go. So think properly and take a wise decision. So moving on to the next question. In an experiment, RBCs were subjected to lysis and unbroken cells were removed by centrifugation. The supernatant was taken and centrifuged at this much. The pellet was extracted with 5 molar sodium chloride solution and again underwent centrifugation. So you have to identify the proteins which you will find in the supernatant. These are the proteins which you can find in the RBC. So which is found in the supernatant? Whether it is band 3, glycophorin, GPCR, spectrin, which one? Which one you will find in pellet or supernatant? Which one you will find in debris? If you are aware of the nature of these proteins, you can get the answer of this question. What should be the answer? What should be the answer? Can we eliminate any option from here? Is it possible to eliminate any option from here? I can see a mixed response like 3232 is coming. Surface protein you are saying, okay. Surface protein in what sense, Anupam? 3 can be eliminated, okay. GPCR, yes, GPCR connecting to RBC is not a relevant thing. So we can eliminate this. Now what you are left with? Band 3, glycophorin and spectrin. So what is the nature of these proteins? Another clue which I can give you is that you have to filter it out which one is an integral protein and which one is a peripheral protein. If you know that idea, you'll be able to get the answer. Which one is an integral membrane protein? Which one is a peripheral protein? Upon centrifugation, who will be settling where? Peripheral or integral? If you are able to get this information, if you are able to find a connecting link over here, you'll get an answer. I can see majority of you are saying 2. Anyone else would like to give your answers? Some miscellaneous response I got. Okay. So let's check it out, the answer. So the correct answer over here is spectrin. Why? All the given proteins are integral proteins of RBC excepting spectrin. And spectrin will be an example of peripheral protein which remains in the supernatant. Remaining all are high molecular weight proteins, integral proteins and all. And they are strongly attached to the membrane. So obviously when they will be undergoing centrifugation, they will be seen as a pellet below. But in the supernatant, some slightly attached proteins you can find, that is spectrin. Okay. And can anyone tell me glycophorin belongs to which category of integral membrane proteins? You are aware that we are having 6 types of integral membrane proteins. So among them, glycophorin belongs to which category? Do you remember? 6 types of integral membrane proteins. Glycophorin belongs to which category? Anyone able to recollect? Unit 2 is something where you can get direct question as well as experimental questions like this so far whatever I had discussed with you. But trust me, this experimental questions will also be easy if you're aware of the options. That means what is the function of that protein. Yes, absolutely correct. Anusha and Surendra, this belongs to type 1 integral membrane protein. Okay. So moving on to the next question. Fluorescently tagged proteins was used to study protein secretion in yeast. Fluorescence was observed in Golgi, secretory vesicles, and rough endoplasmic reticulum. Identify the sequence of events. That means from where to where the fluorescence will be observed. That means how the protein is moving to different compartments. That is what they are trying to say. No, sunshine 5 is a different thing. 5, you are having a lipid anchor over there. It is a lipid linked protein. Okay. So 5 will not come. Yes, Pravalika, that was type 1 integral membrane protein. So what is the answer over here? I mean, the arrangement or the movement of proteins from one compartment to another compartment. This is a part of what system? When we speak of Golgi, endoplasmic reticulum, secretory vesicle, this becomes a part of a specific system. What is that system? Okay. Anusha, Saujanya, Anchal, I can see your answers. Anyone else would like to give you inputs? Very good. We call these components as endomembrane system. Now, if you remember the endomembrane system, try to recollect how the proteins are getting imported from one compartment to other. You are having Golgi, secretory vesicles, and rough endoplasmic reticulum. I guess now you will be able to answer correctly. Yes, Deepshika, Kirtana, Ajay. Vesicular transport. Yes, Kirtana. Anyone else would like to give your inputs over this question? Okay, let me show you the answer. As I can see, majority of you have said the correct answer, that is C, A and B. Rough endoplasmic reticulum, Golgi and secretory vesicle. Now, how does it work? See, always remember this format. The proteins from rough endoplasmic reticulum will be carried to Golgi complex. This carryover is done by transport vesicles. So transport vesicles are responsible for what? Carrying a content or carrying a protein from one compartment to the other. Now from this Golgi complex, it has to be secreted out, either in the form of other compartment, or it will be fused with the plasma membrane, or it will be released outside. So we can say this is a secretion process. And this is carried out by whom? Your secretory vesicle. So that's the reason option 3 is the correct answer over there. Okay. I can see majority of you have given the correct answer, but those who could not give, I hope so you understand how exactly it works. Just remember this order. Rough endoplasmic reticulum, transport vesicle, Golgi compartment, secretory vesicles. Why we are going for secretory vesicles here? Because they are being secreted outside of the cell. Maybe fused with the plasma membrane, maybe combined with some other compartment. Okay, that is the reason. Next question. Membrane proteins are synthesized on endoplasmic reticulum and transported to various organelle membranes. One hypothesis regarding protein sorting is that proteins with a shorter transmembrane portion would partition into thinner membranes. So identify this point. You are provided with 3 observations. What were they? It was found that Transmembrane portions of proteins in Golgi membranes are shorter than those in plasma membranes. Presence of cholesterol increases the thickness of the bilayer. Phospholipid composition of Golgi and plasma membrane remains the same. Now, you have to identify the correct statement from here. Proteins in the plasma membrane have longer transmembrane portion compared to Golgi. The other statement will be the opposite. Longer transmembrane portion present in Golgi compared to plasma membrane. Or both of them are having the same length of transmembrane portion. Cholesterol is present more in Golgi compared to plasma membrane. Now, according to you, what should be the correct answer over here? Bit twisty question, but if you read the question properly, you'll get the answer from the question itself. The answer is there in the question only. Read the question once, twice. In exam also, when you are going through the question, if you are not able to understand in the first attempt, try to read it twice and thrice. Obviously, you will get some point over there. What should be the answer over here? Okay, I can see Kirtana has given the answer. Others? Apart from Kirtana, anyone else? Once again, I am saying the answer is there in your question itself. If you read the question properly, you are going to get the answer. Yes, Sunshine also gave the answer. Just focus on this particular sentence. Proteins with shorter transmembrane portion would partition into thinner membrane. Thinner membrane indicates what? Is it a Golgi compartment or a plasma membrane according to you? We can just imagine what is the function of a Golgi, what is the function of a plasma membrane, the major function related to membrane. So, accordingly, thinner membrane is contributed by Golgi or plasma membrane. What should be the answer? Kirtana and Sunshine are saying thinner membrane contributes to Golgi. Now, okay, Anusha Saujanya also said the same. Now I can see all of you has given the correct answer. See, you only gave the correct answer. I did not said you anything. Just I helped you out in understanding this question. So the correct answer over here is answer 1. If you understand this sentence, if you understand what is a thinner membrane, you can easily click on to the first option. So the proteins in plasma membrane will have longer transmembrane portion compared to Golgi membranes. And obviously, plasma membrane is a selectively permeable membrane, outer membrane, inner membrane, everything is there. So obviously, it has to be very thick compared to the Golgi vesicles, right? So obviously, you can omit that. And if you look, and if you have given this as the correct answers, obviously, these two statements will not make any sense. Coming to the fourth statement, cholesterol is more in Golgi membrane than in plasma membrane. No, cholesterol is present more in plasma membrane compared to the Golgi membrane. Okay, so that's why you have answer 1, statement 1 as correct. Now, apart from that, some information over here. Transmembrane domains of Golgi membrane proteins are generally shorter in length compared to those found in plasma membrane proteins, because your Golgi membrane is a thinner organelle compared to the plasma membrane. And moreover, the Golgi proteins will not span a lipid bilayer. So, the shorter transmembrane domains enable these proteins to anchor within the Golgi compartment while conserving space and ensuring proper functionality. So additionally, what happens? Golgi membrane proteins also contains a high proportion of aromatic amino acids. We are having only 3 aromatic amino acids: phenylalanine, tyrosine, and tryptophan. So these amino acid is known to play a very important role in protein to membrane interaction, thereby contributing to stability and positioning of proteins within the Golgi compartments. So two reasons. One is Golgi is having short transmembrane portion. Second reason, they are having aromatic amino acids. Okay, so that's the reason option or the statement 1 is the correct answer. Okay, this is the way you can approach a question. Next question. Which one of the following statement correctly applies to the proteins which are translated on your RER, rough endoplasmic reticulum? Cytoplasmic proteins are targeted to the nucleus in response to hormonal stimulation. Proteins targeted to lysosomes, plasma membrane, and cell exterior. Proteins that are targeted to nucleus through ER lumen, as the lumen is in direct connection with the intermembrane space of nucleus. All proteins get targeted to peroxisome. So what should be the correct answer over here? That means what kind of proteins are translated on RER? What should be the answer? Now here you need to apply the concept of what? You need to apply the concept of the one which I had said you earlier, that is co-translational transportation and post-translational transportation. If you know the difference between these two, you will be able to get this answer. What should be the answer? Have a think and tell me. Did not get anyone? Do you know the difference between these two mechanisms, co-translational and post-translational mechanisms? Are you aware of these terms? If you are aware of these terms, just correlate over here. Three, two, three. That is the answer I go. Okay, co-wins with ribosomes. That means you meant to say it is related to endoplasmic reticulum, right, Kirtana? Rough endoplasmic reticulum. Post with ATPases. Some confusion over here. Okay. We can see most of you has given option statement 2 as the answer, but still there are someone who are giving option 3 as the answer. So you're completely sure that statement 1 and statement 4 is not required. That means a tug of war between statement 2 and 3. That is correct. Ashu, the concept which you had said me is correct. So definitely it is not going for peroxisome as of now, right? So if you see proteins which are targeted to the nucleus through endoplasmic reticulum, first thing I want to ask you, does the proteins move from endoplasmic reticulum to the nucleus? If so, in which kind of modification? Does it go? Confused between 2 and 3? No confusion. See, even if you have a confusion over here, check out the other sentence. Lumen is in direct connection with intermembrane space of the nucleus. Is it so? Does endoplasmic reticulum, lumen, is connected to intermembrane space, or the ER is connected to outer nuclear membrane? What should be the correct answer over here? Does ER is connected to outer nuclear membrane is correct, or lumen connected to intermembrane space is correct? What should be the correct answer? This one is the correct answer, right? Endoplasmic reticulum is connected to outer nuclear envelope. So obviously, what should be the answer over here? Second statement. Okay. Now, the answer is second. Now, if you see this image, see, this is a co-translational import as mentioned by Ashu, where the translation is going on, but at the same time, destination is also revealed, like where it has to go. So either it will remain in the ER, or depending upon the signal sequences, it will be translocated to Golgi, secretory vesicles, lysosomes, or plasma membrane. So remember, there is no point of nucleus over here. But on the other hand, post-translational import means after the translation only, the protein is being decided where to go, whether it should be present in the cytoplasm or has to be transported to nucleus, mitochondria, chloroplast, and peroxisome. So these organelles participate where? In the post-translational transportation of proteins. So that's the reason statement 1 and 4, even 3 is excluded, because they mentioned about this only, right? They are mentioning about post-translational modification. But our question was from ER where it goes. So it comes under co-translational import. So you get the answer. Okay. I hope so. This concept is also clear to all of you. Thank you so much, Suman. Okay. Next. Now, following the domain organization of 3 proteins that are targeted to mitochondria. So here are 3 proteins which has to enter inside mitochondria. But we know what mitochondria is having different compartments, right? From outer membrane, inner mitochondrial membrane, intermembrane space, matrix. So in all these compartments, protein has to be imported. So what should be the nature of the polypeptide to enter into the specific compartments? So here they have provided with the option what is A, B and C. So you have to identify A goes to which compartment, B goes to which compartment and C goes to which compartment. Find out. Now this will be cleared if you remember, if you have attended the in the class, I had given you the kind of proteins that needs to be imported into different compartments. So every polypeptide is having some specificity towards that compartment. Based on that, this question is given to you. Here you don't have an option, but you have to recollect that image provided in Lodish, that how a specific polypeptide enters into a specific compartment. That's the only way out. Okay, I can see Kirtana has given the answer. Others? See, the first protein will have matrix protease cleavage site. Second polypeptide will have matrix exposure, inner membrane exposure. Then you are having a hydrophobic region. Hydrophobic region is for what? Tethering or attachment to a membrane. Last one is interacting with outer membrane space. Outer membrane import pore. So which portion of mitochondria will be responsible for interacting with outer membrane pore? Okay, I can see some miscellaneous answers are coming up. One, okay, all the options are mentioned by everyone. It means everyone has mentioned 1, 2, 3, 4. So not a single option that is left off. No problem. Try to analyze. You'll get the answer. Analyze in the sense you have to recollect the image. Shall I give the answer? So those of you who have given answer 1, very good. Yes, the correct answer is 1. But those of you who could not give the answer, don't be disappointed because now you are learning. And from here onwards, you will not make the same mistake again, right? In the perspective of transport of proteins. So how this 1 is the correct answer? See, here you are having matrix protease cleavage site. That means it has to enter inside the matrix. No other sequences. Only they are having MTS, matrix targeting sequences. So that's why you are having matrix protease cleavage site. So obviously A goes to the matrix. Second one, that's why I specifically mentioned hydrophobic region means that will be tethered to the membrane. Inner membrane protease site is also there. And they have a brief exposure to matrix also. If you remember, we have discussed 3 different pathways of inner mitochondrial membrane, right? Or among them, 2 will be exposed to matrix and 1 will not be exposed to matrix, directly transferring. So one that exposed to matrix is this one. So the nature will be like this. And the last one is interacting with outer membrane. Obviously, we don't have inner membrane that will be interacting with the outer membrane pore. In between, whatever the space is that is occupied by whom? Intermembrane space. So this will be interacting over there. Okay. So this is how you will be getting the answer. So if you see, this was given in your Lodish book. You may have a check on that. Matrix. That means you will be having matrix targeting sequences. Inner membrane. They have exposure to matrix and hydrophobic sequences is also there. Another pathway. They have matrix exposure recognized by an exporter protein known as OXA1, which will help in tethering the protein to the inner membrane. And the last pathway, it does not have any matrix targeting sequence that indicates what? There is no exposure to matrix directly tethering to the inner membrane. Now, if you take the example of intermembrane space, what happens here? Intermembrane space, you will have one as a matrix targeting sequence and second as an intermembrane space targeting sequence. So that polypeptide contains 2. Okay, so it will be crossing the inner membrane as well in that case, because if it has to reach matrix, definitely it has to cross inner membrane. Then another pathway where it will enter inside the intermembrane space via general import port. That means here there is no interaction or connectivity between inner membrane or the matrix. And outer membrane, if the protein is having outer membrane localization sequence, it will be tethered over there. So no need to expose to inner membrane, intermembrane space or the matrix. Okay. I hope so this question is clear to everyone. So we are stopping it over here. Remaining questions specifically from cell protein targeting and cell organelles we will be discussing in the next session. Okay. So stay updated and go through the sessions and go through the classes so that you'll be able to give the answers better. Okay. Thank you so much.